the later what it showed that most of the vitamins, they did not contain amino group. Okay. Now definition. Now the definition of vitamins are that they are non-caloric organic nutrients occurring in different natural foods and needed in small amounts like in microgram and milligram per day, and they are essential for growth, for proper growth and maintenance of our good health. Please excuse me, there is some technical error. I'm not able to move my slides. Okay. You have to click here. Okay. Just click here. Okay. So, okay, this is the definition of vitamins. Now, we have seen that these vitamins, they are nutritionally very important, okay, for the proper growth and for good health. But in clinical practice, sometimes their role is overemphasized. Why? Because the vitamins, they can cure only a disease which is being caused by its deficiency. Okay. It can occur when there is a reduced intake, where impaired absorption, impaired metabolism, increase in demand, or there is a there are increased losses. Okay, so this you have to remember that if the vitamins, they can be given as supplements only the diseases which are being caused by their deficiency. Okay, now sometimes when it is being given, they are being given in as supplements, okay, in higher doses, but for a very long time, some of the vitamins, instead of boosting up the health, instead they cause toxicity in our body. Okay, and especially the vitamins which are fat soluble, because one, they are not readily excreted by our body, and number two, they have an affinity to get stored in the body, so they can cause toxicity. Okay. Now the vitamins like fat soluble vitamins A, D, and K, these vitamins, they can store in liver, whereas vitamin E, it gets stored in adipose tissue. Okay. As they are being stored, so they need not to be taken in daily in diet. And we have already seen if too much of it consumed, so they can also reach to the toxic levels. Okay. Sorry for the interruption, students. Actually, there is some problem. Starting something. Please press mute करो. Okay. Now, before starting the fat-soluble vitamins, I just wanted to say this, that fat-soluble vitamins, how they can be differentiated with the water-soluble vitamins. The water-soluble, see, as they are fat-soluble, so for their absorption, they would be needing as they are being absorbed as other dietary lipids are absorbed. Okay, so their absorption is just like the absorption of other dietary lipids, whereas the water soluble vitamins, they do not, they just directly get absorbed through the intestinal cell. Okay, number two, as they are lipid soluble in nature, so they need to have a carrier, okay, to be transported in the blood, whereas water soluble, they do not want, they do not have a do not have a carrier to be transported in blood. Okay, then number three is the excretion. We have seen that lipid soluble vitamins, they are not readily excreted in the excreted through the body, whereas water soluble, they can very readily excreted through the body. The number four is that if lipid soluble vitamins, they can be stored in body, whereas water soluble vitamins, they are not stored in body. Then because as if they are being consumed in excess, and because they are not being readily excreted, so they have this affinity to get deposited, to get stored in our body, and can cause toxicity. So with vitamin with the fat soluble vitamins, if they have been taken in excess, they are very likely to cause toxicity, whereas water soluble vitamins, they do not cause toxicity. Okay, so as we have a store of vitamin D, fat soluble vitamin D stores in our body, so they need to be taken in periodical, in weeks or months, whereas water soluble, as they cannot be stored, they need to be taken after every second or third day. Okay, so their intake is more frequent than the fat soluble vitamins. Okay, so now we will be discussing the fat soluble vitamins or lipid soluble vitamins, vitamin A, E, D, and K. Okay, so starting with the vitamin A. Okay, now this vitamin A, it is being generic term which is being given to all the compounds from animal sources exhibiting the biological activity of vitamin A. Now vitamin A, they are chemically, they are polyisoprenoid compounds containing a cyclohexenyl ring in them. Okay, so the vitamin A chemically, in fact, all the fat soluble vitamins, they are basically the derivatives of isoprene molecule. Okay, so this vitamin A, in addition to having this polyisoprenoid unit, they also have a cyclohexenyl ring, which is also called beta ionol ring. Now about the animal sources, like from animal sources, if we take vitamin A, in them the vitamin A exists in the form, it is existing in active form, that is retinol. So actually retinol, it is the active form of vitamin A in our body, and most of the actions, they occur through this form, either in the as such in retinal form, or it get converted into different derivatives like retinol and retinoic acid to exert its different actions on different peripheral on its different target tissues. Okay, so animal sources, through animal sources, we usually take vitamin A in its active form, that is retinol. In plant sources, it usually it is in the plant sources, like we have the vegetables like carrot, papaya, mango, these all yellow color, they have a pigment in them, yellow pigment, which is very rich in beta carotene. So the beta carotene is the form of vitamin A that we get from the plant sources. Okay, but this beta carotene, it is a precursor form that we get from plant sources. It is a provitamin and it consists of two molecules of retinol. Okay, and these two molecules, they are being joined together through their aldehyde groups. So in order to for its to act in our body, so it needs to be cleaved into two retinal molecules. Okay, so from plant sources we get vitamin A in the precursor form, that is the beta carotene. Now beta carotene, it is only 1/6 as effective as a source of vitamin A as retinol, because it is not being efficiently metabolized our body to vitamin A. Now there are two terms, retinoids and carotenoids. The retinoids, they are basically the natural forms of vitamin A that we get from the plant or the animal sources, and also it includes the synthetic analogs of retinol. So these all, they together constitute the retinoids. And the carotenoids are the compounds which are having the beta carotene like activity. Okay, so we call them as a carotenoids. All the compounds or the organic compounds which have a beta carotene like activity, they are being called as beta carotenoids. Okay, now coming to the structure. Now structure, as we need to know the structures just to understand the function of the vitamin A in our body. Okay, so as I already told you that the active form of vitamin A in our body, it is retinol. Okay, but through other forms also like retinoic acid, it exerts its actions on its specific target tissues. Okay, so retinol, you see here, you can see this is an isoprene molecule. I told you that isoprene molecule, this vitamin A, it is a derivative of isoprene molecule. So this is 1, 2, 3, 4 carbon atom, 4 carbon atom isoprene molecule, and this is, here you can see two isoprene molecules, they are being attached together, to which there is a cyclohexenide ring. Okay, attached. So this molecule, it is retinol. Just to understand, you don't have to remember these to learn these structures. Just for understanding this mechanism of action of vitamin in body, you need to know these different forms. Now these different forms, they are also called retinoic acid are also called vitamins. Why we call them vitamins? Because they have a similar structure, chemical structure, and but they have a similar structure with qualitatively similar physiological function. Okay, so we call them as vitamins. So in retinol, you can see on this end we have a we have a hydroxyl group, so we call it as a retinol. When it is substituted by aldehyde group, it becomes retinol. And again here you can see there is a carboxylic group, so we call it as a retinoic acid. Okay, so these are the structures of different forms of vitamin A. Okay, then beta carotene, here you can see the beta carotene, actually they are 1, 2, 3, 4. Okay, this is one unit having the retinal structure. Okay, and this is another one, 1, 2, 3, 4. Okay, so there are two molecules of retinal which have been joined together through this bond. Okay, so the beta carotene, it consists of two retinal molecules. Okay, now coming to the absorption. How does absorption occurs? Now in animal sources, we from the animal food, as we have seen, the vitamin A, it is in the form of retinoid esters. Okay, so being lipid soluble, it would be needing the bile droplets to get dispersed in the intestinal lumen. Okay, and then this in intestinal lumen, this retinol ester, it is being hydrolyzed by a hydrolase enzyme into retinol and a free fatty acid. Okay, then this retinol, then it is absorbed directly through the into the intestinal cell. The inside, what happens in intestinal cell, because it is lipid soluble, it need to be packed into chylomicrons after re-esterification to be released into lacteals or into the circulation. Okay. Now, what happens to the vitamin A that we have taken with the plant sources? And we know that in plant sources, vitamin A is in the form of beta carotenes. So what happens? Beta carotenes, these compounds, they get absorbed as such through the intestinal, through the mucosal intestinal mucosa inside the intestinal cell. Okay, then what happens in the intestinal cell? It gets cleaved into, because we know that this beta carotene, it is in a precursor form in plant foods. Okay, so it needs to be converted into the active form. So it happens what to make it happen, in intestinal mucosal cells, they get cleaved into two molecules of retinal by an enzyme beta carotene dioxygenase. Okay, now this has to be followed by the reduction so that the retinal form will get reduced into retinol. Okay, so finally from beta carotene, two retinol molecules are produced in the intestinal cell. Then they get incorporated into the chylomicrons, to be chylomicrons after esterification of with the saturated fatty acids, and then they released into the lacteals to enter into the systemic circulation. Okay, so this is a diagram just to make you understand how the beta carotene, it is being hydrolyzed inside the intestinal cell. Okay, so it happens with the help of bile salts. It gets absorbed through the intestinal cell, and inside the intestinal cell, with the action of beta carotene dioxygenase enzyme, it gets cleaved into two retinal molecules. This is one retinal molecule, and this is another retinal molecule, or you can say retinal aldehyde. Okay, then what happens? You can see by this, with the help of retinal reductase enzyme, it gets converted into retinol. Okay, and here the coenzyme is NADPH, and or it can also be converted into retinoic acid. Okay, this will be in the presence of an enzyme dehydrogenase. Okay, and here the coenzyme is NAD. So, okay, now here you can see in retinoic acid, in retinol, or in the retinal, these double bonds, they are on the these double bonds, they are basically in a trans configuration form. So we can also call this retinal molecule as trans retinal. So that from trans retinal is formed through retinal reductase enzyme, and from trans retinoic acid can also be formed, that is through the enzyme dehydrogenase. Okay, so you should be knowing about these interconversions, because you need to understand, in order to understand the mechanism of action of vitamin A, you need to know about the different forms of vitamin A. Okay, so and also should be knowing either in this trans form or it is in the cis form. Okay, now after absorption, we have already seen that they have been incorporated into chylomicrons. Okay, and they, through the lacteals, they reach the systemic circulation. Here they have been distributed to the tissues having the lipoprotein lipase activity, and get converted by its action, get converted into the chylomicron remnants. Now these chylomicron remnants, then they've been taken up by the liver, okay, along with the retinol. Now in liver, what happens? Either depending upon the need of the body, right? Either they will be transported to this retinol will be transported either to the target tissues for its actions, or it can also get deposited in liver as in the storage form. Okay, so in liver, it is being stored in the form of retinol ester. Okay, and mostly the fatty acid which is being esterified with this is the palmitate in the form of retinol palmitate. Okay, and where the cells are the perisinusoidal stellate cells, we also call them the lipocytes. Okay, so in these cells they get stored. Okay, the vitamin A get stored in the form of retinol ester. Or else if it is being transported, it need to be transported to the target tissues. Being lipid soluble, it need to be then carried in the blood through a protein carrier. So it need to bind to some protein carrier. So what happens in hepatocytes? This retinol then it binds to aporetinol binding protein. Okay, and then they form a hollow retinol binding protein and get released into the systemic circulation to reach their target tissue. Okay, so this is how once it is being released into the blood to be delivered to the target tissues. So what happens to the retinol now, which is being bound to the retinol binding protein? It is being in target tissue. It is being taken up by there are receptors on the target tissue cells. Okay, so which take up the retinol inside through the internalization, but they do not take the retinol binding protein. So what happens to retinol binding protein? Retinol binding protein returns to the circulation, and retinol it internalized into the target tissue cell. Okay, and where these all cell receptors for retinol binding protein are present, it is in eye, skin, gonads, and other tissues. Okay, so once now it is inside the target tissue cell, so again it binds to this retinol. It binds to cellular retinol binding protein. Okay, which is basically the receptor for this retinol, and then this retinol along with a cytoplasmic receptor, it is being transported inside the nucleus, and then this complex it attaches itself to the hormone response elements of the DNA, and thus it influences the expression of certain genes which are dependent on vitamin A. Okay, so basically what it does to the genes, either it causes derepression or induction of these genes, and due to which specific proteins they are synthesized, and through these and through the synthesis of these proteins, the vitamin A it acts on its target tissues. Okay, now this is a diagram which is basically showing the overall metabolism of vitamin A. So here you can see from the animal food, it is being taken, vitamin is taken inside in the form of retinol esters, and from the plant source it is in the form of beta carotenes. Okay, अब यहाँ पे human में क्या हुआ, intestinal human, this retinol ester it is being hydrolyzed by the hydrolyzers into retinol and free fatty acid, and retinol it get absorbed to the brush border of the intestinal cell and it goes inside. Okay, here it comes here. Now what happens to beta carotenes? As they are lipid soluble, so they can easily diffuse through this surface, through the luminous surface of the intestinal cell, and it gets inside the inside the intestinal cell. Now what is happening in the cell? This retinol, okay, this beta carotene, it is being acted upon by dioxygenase enzyme, get converted into two molecules of retinol. Then by the enzyme reductase, it is getting converted into retinol. Now this retinol, either from animal source or from the plant source, this retinol it esterifies again with this free fatty acid. Okay, and then retinol ester is formed. Now this retinol ester then get incorporated into chylomicron. Chylomicron is the intestinal cell into the lacteals, and through the lymph it reaches the blood circulation. And in blood circulation, you can see here it is in the retinol ester form. Okay, now this after being acted upon by the lipoprotein lipase enzyme in different peripheral tissues, this chylomicron gets converted into chylomicron remnant, and this then having retinol ester, it is being taken up by the hepatocyte. Okay, and inside the hepatocyte, this retinol ester again get hydrolyzed, and this retinol is released. Then this retinol, being lipid soluble, it need to have some carrier in the blood. Okay, some protein carrier, so it binds to the apo retinol binding protein to form the holo retinol binding protein, and then it is being released from the hepatocyte to the systemic circulation. Okay, from the blood circulation. Okay, through blood, it reaches the target tissue. Okay, this is the target tissue, and here we have the retinol binding protein. Here is a receptor for this protein. So what happens? This receptor internalizes the retinol inside the target tissue cell, whereas this returns back. This retinol binding protein returns back into circulation, and then it exerts it. Then it binds to the cellular retinol binding proteins, and then it forms a complex, and this complex goes to the nucleus, and there they bind to HRE of DNAs, and then influences the expression of certain genes which are dependent upon vitamin A. Okay, so this is the overall view of the metabolism of vitamin A. Now, different forms of vitamin A we have already discussed. Vitamin A in our body can act through different forms, like that is retinol, retinal, or retinoic acid, and all these forms, they have a unique biological functions in a body. Okay, now retinol and retinoic acid, see, as they act like steroid hormones. So their mechanism of action is just like steroid hormones. As we have seen, they have the cytoplasmic receptors in them. Okay, so now these receptors, why we say that they act like steroid hormones? Because the receptors through which they act, they are the members of the same receptor superfamily to which the receptors of steroid hormones belong. Okay, now what is the function? We will do in our next slides. We see what different functions, different forms they have in our body. But just to enumerate here, I have you can see this that the retinoic acid, basically in its trans retinoic acid, it is being responsible for promoting the cell growth development and for the differentiation. Okay, it is responsible for promoting cell growth and cell differentiation. Okay, and by this by this action, it helps in maintaining the different epithelial linings in our body. Okay, we'll see later on, and also through this action, it influences our immune system. Okay, then retinoic acid. Okay, this is about the retinoic acid. Then retinol. What is the function of retinol in our body? Trans-retinol. It supports the reproductive system by maintaining the germinal epithelium in gonads. Okay. Then retinal in the form of 11-cis retinal. It has a role to play in vision in poor light. Okay. So now, before starting the before starting the different functions that vitamin A exert in our body, I would like to again tell you how the different interconversions of these different forms of vitamin A is happening in our body. Okay, in different target tissues. Like retinol, as we know, it is an active form from the both plant and animal sources. We have taken the vitamin A in the form of retinol. Now, this retinol can be converted as retinol. Now, this retinol can be converted into retinal. Okay, it is in the presence with the help of enzyme dehydrogenase, where we have already seen that with the help of enzyme dehydrogenase, where NAD is the coenzyme. Okay, reverse can also happen. We have seen when the beta carotene was getting converted into two molecules of retinol and finally getting converted into retinol, so the enzyme required was the reductase. It was reductase. Okay. So then this retinol can be converted into retinol with the help of this enzyme reductase. Okay. Then retinol can be converted into retinoic acid. Again, it will be in the presence of an enzyme dehydrogenase. Okay. So this is another diagram which is showing the different interconversions of different forms of vitamin A. Okay. So here you can see from retinol, retinoic acid can be formed with the help of dehydrogenase. Then from alltrans retinal, retinoic acid can be formed because it is getting oxidized, so enzyme dehydrogenase is required. Okay, and here from alltrans retinal with the help of enzyme reductase, it can be converted into retinol. Okay, here because here the reduction is taking place, aldehyde group is getting converted into the hydroxyl group. Okay, and then there is one more interconversion. All trans retinal can be converted into 11-cis retinal. Okay, so enzyme, because these are the cis and trans, they are the isomers. So the enzyme which is being required would be the isomerase. So all trans retinal can be converted in the presence of enzyme isomerase into 11-cis retinal. So this 11-cis retinal will be required is responsible for vision in dim light. Okay, it helps in vision in dim light. Okay, now we would be taking individually all the actions of vitamin A in different forms in our body. So we'll be starting with the role of vitamin A in vision. Okay, now it has been discovered by Wald in 1967 that vitamin A was responsible for vision in dim light. Okay, and for which he was also awarded a Nobel Prize. Now I think you all be knowing that now in our for the vision, they are in our eye, they are photoreceptors, okay, which are responsible for our sight. Okay, so and these photoreceptors, they are present in retina, and they are usually, these photoreceptors are being placed beneath the retinal pigment epithelium. Okay, so there are two kinds of photoreceptors in retina. One are rods and second one are cones. Okay, now rods, they are responsible for the vision in poor light or dim light, whereas cones, they are responsible for color vision or for the vision in bright light. Okay, now we would be discussing now the function of rods. Okay, now rods, they basically, how do we see in dim light through rods? It is just because of the presence of protein in them, that is a rhodopsin. Okay, now this rhodopsin, it is a protein opsin, okay, to which is bound the vitamin A, and in this the form of vitamin A is 11-cis retinal. Okay, so always remember that this is happening, we are able to see because of the vitamin A in the form of, it is 11-cis retinal. And okay, so now before starting the mechanism how the rods, they are responsible for the vision in dim light, I just want to I would like to know you people that what is the structure of, you should be able to understand what is the structure of a rod cell in retina. Okay, now this is a rod cell. Okay, ये rod cell है. इस rod cell, it has two parts. One is the outer segment and it is and another one is the inner segment. Okay, now in outer segment you can see these the structure. These are basically are the discs. In these discs are present the visual pigment rhodopsin, okay, which is responsible for the vision in dim light. Okay, now you can see here on this on cell membrane on this rod cell, there are two pumps. One is a sodium potassium ATPase pump. The second was a cyclic GMP dependent sodium potassium channel. Okay, so there are two ion channels in its cell membrane. So what this does, when this rod it is in the unstimulated state, or when there is no photon of light, it is falling on the rod cell, then what happens, the potential across the rod cell, it is it remains in the depolarized state. Do remember that it remains in the depolarized state. How does it remain in the depolarized state? Now the cyclic GMP depolarized state means the potential across to be, it should be potential uh, there should be a positive potential across the cell membrane of the rod cell. And how does it happen? It happens with the help of cyclic GMP dependent sodium potassium channel. Okay, now when it is unstimulated, when there is no photon of light passing on the rod cell, so what happens, the cyclic GMP levels, they are very high inside the rod cell. So what they do is that when the cyclic GMP levels, they are very high, they keep this channel open. Okay, so due to which sodium and sodium and calcium channels can go inside and can make this potential and generate a positive potential across the cell membrane of the rod cell. Okay, so due to which it remains in the depolarized state. Okay, so now what happens when it gets stimulated? After stimulation, rhodopsin, when a photon of light, it falls on the rod cell. What it causes the bleaching of the rhodopsin pigment which is present in these discs. Okay, so bleaching, and once this bleaching has occurred, then this bleaching, it leads to change in the permeability of the rod cell, due to which these channels, they close. These cyclic GMP dependent sodium calcium channels, they close. And because of the closing of these channels, what happens, because of this pump, sodium potassium ATPase pump, the potential across the cell membrane of the rod cell, it becomes hyperpolarized. Okay, so once they become hyperpolarized, it triggers a generation of the generation of nerve impulse in the rod cell, and once it is being generated, it leads to this nerve impulse. Then from these rod cells, it goes to the bipolar cells, then it gets transmitted to the ganglion cells, then to the optic nerve, and finally it goes to the optic center in the brain for the visual processing. Okay, so in this way, we are now able to see in dark light. Okay, so now how this we have seen that a change when a photon of light, it falls, or when a photo excitation occurs of these rod cells, so a photon of light is there, then we have seen that the bleaching of rhodopsin, it occurs. Okay, so bleaching, it means that this rhodopsin, it through various intermediates, it gets converted into meta-rhodopsin. Okay, so during this conversion, what happens, the 11 cis retinal, which is being bound to the rhodopsin, it gets converted into all-trans retinal. Okay, so here you can see 11 cis retinal at 11th position. You can see that here you can see that it is in the cis position. Then what happens once the photon of light it hits the rod cell, then this 11 cis retinal it gets converted into all trans retinal. Okay, so what happens because of a change in the structure of this 11 cis retinal, which is bound to the opsin protein of rhodopsin, it causes a conformational change. It causes a conformational change in the structure of uh, rhodopsin protein. So once there is a conformational change, the 11 cis retinal it get dissociated from the rhodopsin, okay, from the rhodopsin, or you can say from the meta-rhodopsin 2, it get dissociated, and this opsin this opsin due to its conformational change, what is that it does, it activates a G protein which is present uh, in the cytoplasm. Okay, it activates the G protein uh, which is otherwise is present in a inactive state. Okay, so conformation change is there. There is an activation of a G protein known as transducin. Okay, and once it gets activated, so this GTP which is being bound of this transducin, it is a heterotrimer. Okay, it has 3 subunits: alpha, beta, and gamma. So once this get activated, the GTP which is bound to the alpha subunit of transducin, it get replaced by GTP. Okay, now this GTP what it does, now once it is being bound to GTP, this GTP bound alpha subunit now get dissociated from the protein, this transducin protein. Okay, so you can see here this beta gamma, these subunits they get dissociated, and this then further this gamma alpha subunit having GTP in it, it then goes and then it attaches to the enzyme phosphodiesterase, okay, which was also in the inactive state earlier because of this inhibitory unit which is bound to the phosphodiesterase enzyme. Okay, so once this alpha subunit of transducin or the G protein it binds to phosphodiesterase, it makes it active. Okay, so once it becomes active, now what is the function of phosphodiesterase? It converts cyclic GMP into GM. Okay, so due to which there is a decrease in concentration of cyclic GMP, leading to the closure of sodium channels, which further leads to hyperpolarization of rod cell membrane, and which finally leads to the generation of nerve impulse. Okay, so you should be knowing about what all biochemical reactions they are responsible for the uh, for the vision. Okay, for how this rhodopsin it helps in seeing the in vision in dim light. Okay, so it occurs through a sequel of biochemical reactions. Okay, fine. So once again, I'm just repeating it that when a photon of light it falls on the rhodopsin present in the rod cells, it bleaches the rhodopsin, converts into the meta-rhodopsin 2, due to which the 11 cis retinal which is present in the rhodopsin gets converted into all-trans retinal, and um, this all-trans retinal, since it has been bound to the rhodopsin, because of the change from cis to trans, it causes a conformational change in the opsin protein, and due to which conformational change, there is a dissociation of opsin and 11 cis retinal. Okay, now this then this opsin protein, then it causes the activation of G protein transducer, which with the help of its gamma subunit, with alpha subunit, it activates phosphodiesterase enzyme, and due to which activation of phosphodiesterase enzyme, cyclic GMP is converted into GMP, and decrease in GMP leads to closure of sodium channels, and which leads to hyperpolarization and thus the generation of nerve impulse. Okay, so this is how a nerve impulse it is generated when a photon of light it falls on the rod cells. Now the cell, because of the falling of the light, it has it gets stimulated. Now there are some, it need to go back, but there has to be a reversal of the stimulation also, you know, it has to be terminated. Now nerve impulse which has been generated now need to be terminated, so that it will become, the rods again they become receptive for the next photon of light. Okay, so how does this termination of visual nerve impulse it occurs? Okay, in the rods, it is like where the photon of light, on one hand it is basically what is we have seen in this diagram, it is activating the transducin protein. Okay, but at the other hand, because of the photo excitation or because of the falling of the photon, what is happening, the GTPase activity of this transducin molecule is also getting activated. Okay, so so how this termination will occur? Now this, at the same time, this GTPase activity is also activated. So what this GTPase will do, this will replace this GTP to GTP. Okay, once this GTP replaced by GTP, this alpha unit will bind to the to its other subunits beta and gamma, and thus the stimulatory effect of this alpha subunit it goes off, and this phosphodiesterase activity of this enzyme it stops. Okay, and it gets inactivated. Phosphodiesterase enzyme again becomes inactivated, so due to which there is an increase in cyclic GMP levels, and then which is leading to the opening of sodium channels, and again the rod cell it becomes depolarized, and the nerve impulse generation it stops. Okay, so this is one of the mechanisms by which the termination occurs. Another is that we have seen during stimulation what happens, that 11-cis retinal it has been dissociated from the rhodopsin. ठीक है, तो अगर suppose इनका reassociation हो जाता है, again if it gets reassociated, this 11-cis retinal gets reassociated with the opsin to reform rhodopsin. It also terminates the nerve impulse generation in rod cells. Okay, now how this regeneration of all trans retinal into 11-cis retinal occurs? It occurs either in liver or in retina. Okay, and it occurs because it is a cyclic thing, so we call this cycle as a Wald's visual cycle. Okay, so Wald's visual cycle is responsible for regeneration of all trans retinal into 11-cis retinal. Okay, all trans retinal into 11-cis retinal. So we have seen that when the light falls, rhodopsin it gets in rods it gets bleached. Okay, through different intermediates, finally it forms the metarhodopsin 2, and then this metarhodopsin 2 is formed. This all trans retinal which is being formed during the bleaching process. Okay, so this gets dissociated from the opsin. Okay, here this is opsin, so it gets dissociated. Now what happens? What happens to this all trans retinal? Now this all trans retinal, now it comes into the circulation, into the blood, then through blood it goes to liver. Now this all trans retinal with the help of enzyme alcohol dehydrogenase in an hepatocyte, it gets converted into trans-retinal with the help of an coenzyme NADH. Okay. So here the enzyme responsible is alcohol dehydrogenase for converting this all-trans retinal into trans-retinal. Then with the help of enzyme hepatic isomerase, it gets converted into cis-retinol. Okay, so this is trans-retinol and it is isomeric form. So hepatic isomerase, it gets converted into cis-retinol. Now this cis-retinol, then with the help of dehydrogenase enzyme, because alcoholic, this hydroxyl group is converted into aldehyde group. So here it is getting what? It is getting oxidized. Okay, it is getting reduced. Sorry, it is getting oxidized. So what is happening here? The coenzyme is NAD, so it is getting converted into NADH. So this, once this 11 cis retinal now formed in liver cells, now this will be released into the bloodstream, and from the bloodstream it goes to the retina, and then this 11 cis retinal, it associates again, reassociates with opsin to form rhodopsin. Okay, so this cycle will keep on going for regeneration of rhodopsin. Okay, so this cycle we call it as a Wald's visual cycle, which is responsible for regenerating all trans retinal into 11 cis retinal. Okay, now we have seen that after regeneration, 11 cis retinal, it reunites with the pigment, but sometimes what happens, a little of the vitamin A is destroyed each time. Okay, in this reaction. Okay, so it means that a new fresh vitamin A must be replenished, should be replenished for that small loss. So in vitamin A deficiency, what we see is that patient, the first indication in vitamin A deficiency, or when the vitamin A deficiency or supply is running is low, then a person complains of night blindness. Okay, we call it as a nectalopia. Okay, and there is also an increase in dark adaptation time, because in that case, if the supply of vitamin A is low in our body, or the levels are low, so this small loss which has been has occurred in this regeneration will not be replenished. Okay, now I have just mentioned dark adaptation time. Now, what is this dark adaptation time? Now you have noticed many times that when we shift from bright lit room to a dark lit or the dim light area, first of all we have a difficulty in seeing. Okay, so we are not able to see what is around us, but after a few seconds this vision improves. Okay, so इसका एग्जाम्पल बहुत अच्छा है कि when we enter into a cinema hall from a brightly lit area into a cinema hall, so for some seconds we are not able to see what is around us, but after some time our vision improves. So this time which eye takes to get adapted to the dark is known as dark adaptation time. Okay, and what is this time? In this is the time actually when the synthesis of rhodopsin it is taking place, because now we are entering from the bright light to a dim light. Now even the bright light, when there is a bright light is having the photon of, it is having the photon of lights. Bright light में भी तो होंगे, so those photons of light, they will bleach the rhodopsin. Okay, so it needs to be resynthesized back. So this is the time it takes to resynthesize rhodopsin back. This, we call it as a dark adaptation time. Okay, so definitely if there is a vitamin A deficiency, there would be a reduced stores of the rhodopsin in the rod cells. So in that case, this dark adaptation time would be increased. Okay, so dark adaptation time would be increased. So this is all about the vision of vitamin A in that role of vitamin A in uh, vision in dim light. Okay. Now next is what are the other biochemical functions of vitamin A? See, other they include gene regulation. Okay. Now vitamin A, it also regulates the gene expression of certain genes, specifically those genes which are being responsible for promoting cell growth, development, and differentiation. Okay, and this gene regulation, it occurs through its all-trans retinoic acid and 9-cis retinoic acid forms. Okay, and through this action, basically through regulating different genes, certain genes, vitamin A, it maintains the normal epithelial tissue in our body. Okay, various epithelial tissues, it maintains the still epidermal layer. Okay, it maintains the epithelial lining of gonads, and mucosal epithelial lining of intestine, urinary bladder, or urinary tract. Okay, and as we have already discussed, the action of vitamin A for regulating the gene, for regulating the expression of different genes, it is similar to what the steroid hormones they have. Okay, vitamin A, it acts through the cytoplasmic receptors, which then this complex of vitamin A and cytoplasmic receptor, then it goes into the nucleus and then it binds to HRE of hormone responsive elements of DNA and then influences the gene expression. Okay, now through its regulation on specific genes, on its action on regulating the expression of different genes, vitamin A, it helps in the earliest processes of embryogenesis, and it also helps in the differentiation of 3 germ layers, and also helps in the organogenesis. Okay, because basically it is promoting the cell growth. Its function is to promote cell growth, development, and differentiation. Now, through its retinoic acid form only, vitamin A also mediates, it has a function in immune system. How does it does? It mediates the differentiation of T cells into the regulatory T cells. Okay, so it helps in mediating the differentiation of T cells into regulatory T cells. So it has been seen that in vitamin A deficiency, there is a decrease in hematopoietic cells, stem cells. So why it is so? There may be some, because of the deficiency, there could be some impairment in the cell differentiation. Then we have seen that vitamin A also supports the reproductive system. Okay, so basically, how does it support the reproductive system? Basically, by maintaining the germinal epithelium in gonads. It has been noticed in some cases, because of the vitamin A deficiency, there could be miscarriages in females, or there could be sterility in males. Now, what is its effect on skin? This vitamin A, to its form retinoic acid, it maintains the normal skin health, okay, by switching the genes which are responsible for differentiation of keratinocytes into mature dermal cells. We have seen that basically vitamin A, it is promoting the cell growth and differentiation. So in skin, what is its role? Basically, it switch on those genes which are responsible for differentiating keratinocytes present in the epidermis into mature epidermal cells. So what we have seen is in vitamin A deficiency, because of the deficiency, there is an alteration in skin epidermis, so which makes this skin very prone for the generalized skin infections. Okay. Then in miscellaneous functions of vitamin A, see beta carotene, it also possesses an antioxidant property. Okay. So this antioxidant property, as you all I think have read in lipid in lipids, that these lipid peroxide radicals, they are being continuously being formed in our cells, by various free radicals. So these now free lipid peroxidal radicals, they need to be radicals, they are also being very reactive, so they can damage the cellular proteins, DNA and other lipids, so their action needs to be quenched. So how it is being quenched? With the help of various antioxidants which are present in our body. So beta carotene is one of the antioxidant. It provides it defends against the lipid peroxide radicals. Okay, and basically this acts as an antioxidant in those tissues in which there is a lower oxygen concentration. Okay, and how it does? It takes up the free radical from the lipid peroxide radicals, and takes up and itself gets gets and convert them into the stabilized conjugated alkyl radicals. Okay. Then it has been seen that vitamin E, it is also a very powerful antioxidant, okay, having in our body. So it basically beta carotene, it complements the action of vitamin E, because vitamin E, it is an antioxidant. It has its maximum antioxidant activity when the concentration when there are very higher oxygen concentrations in the cell. Okay, so these two, beta carotene and vitamin E, they have a complementary actions. Okay, so this diagram basically is showing, this is a lipid peroxide radical. Okay, and this is beta carotene. So it has taken up a free, see this is a free electron, you can say here in this. So it has taken up this and converted into a, itself it gets converted into a stabilized alkyl derivative. And this now alkyl derivative is less reactive as compared to this lipid peroxide free radical. Okay. Now coming to the different deficiency manifestations of vitamin A. So what all clinical findings would be there if there is a vitamin A deficiency. Now, as we have seen before in the slides of the role of vitamin A in dim vision, we have seen there that because of the deficiency, there could be night blindness. Okay, because it has a prime role in the vision. Vitamin A has a prime role in vision in dim light. Okay, so in deficiency, so the first indication of the vitamin A deficiency would be night blindness. Okay, and it occurs only when the stores in our body, they are nearly exhausted. Okay, only then it will appear. So the night blindness would be the first indication of vitamin A deficiency. Now in children, there could be this vitamin A deficiency would be manifested like initially as failure to grow. Okay, there would be because the vitamin A is assisting growth of bones and teeth. So it could be the first, in children could be the first indication of the vitamin A deficiency. Okay, now what happens in this, in night blindness, there would be a defective night vision, the visual acuity would be diminished. Okay, and the patient will not be, the person will not be able to read or drive at night. Okay, and we have already seen there would be an increase in the dark adaptation time. Okay, now at this stage, if it is not being intervened by the supplementation, vitamin A supplementation, the further depletion of vitamin of A in our body can cause the ocular changes. ठीक है, तो क्या ocular changes आती है? It can lead to dryness of conjunctiva, which is we also known as xerosis, wrinkling of conjunctiva. Okay, this why it is occurring because of the deficiency of vitamin A, the epithelium of conjunctiva will not be maintained. Okay, and here also the cells which are the goblet cells which are responsible for keeping the eyes moist, they will not there will be a reduced secretion from these cells, okay, because of the deficiency. So there could be dryness of conjunctiva, wrinkling of conjunctiva, and because of the because that normal epithelium is not able to be maintained, so keratinization of epithelium will occur, because of which there could be thickening of the conjunctiva at some places, and these thickening they will be seen as Bitot spots, okay, as Bitot spots. Now if this dryness of conjunctiva spreads to the cornea, it makes the cornea, the corneal epithelium, it becomes lustrous and glassy. Okay, and further, if the epithelium it becomes keratinized, so it will lead to the condition we call it as a xerophthalmia. So first, night blindness, if not treated, can lead to xerophthalmia, and if xerophthalmia persists for a long time, it can then cause the keratomalacia, which is known as which is the what is keratomalacia? It is the corneal softening. The cornea becomes very soft because of the degeneration of the epithelium. Because in deficiency, this epithelium, corneal epithelium, is not being maintained. Okay. So later on, because of the softening, cornea becomes very prone for the bacterial infections, which can further then lead to corneal opacities, ulceration, and perforation, and finally the blindness. Okay, so you should know this sequelae of vitamin A deficiency if it is not being intervened in between. So it can lead to different it then passes through different stages, and ultimately it can lead to blindness. Okay, the first is night blindness. If not treated, can be xerophthalmia, then keratomalacia, and if not treated, then finally the person may become blind. Okay, now it has been seen that the most common cause of blindness in children below 5 years is the vitamin A deficiency, and which is preventable. If it is being diagnosed at a very early stage, it can be prevented. ठीक है, तो उसमें अगर suppose होता है तो we can give this vitamin A as prophylactically to these children. Okay, and it usually being given in a single dose, and to the children which are below 1 year. Now this diagram, it is showing the Bitot spots. Here the keratinization of conjunctival epithelium has occurred, and it will appear like this. Okay, so this is the Bitot spot. Then keratomalacia, I think you will not able to appreciate much through this picture. It is showing the softening of cornea and vascularization, okay, in cornea, leading to the opacity, corneal opacity, because of the various bacterial infections. Okay. Okay, this was all about the ocular changes. Now, what changes will occur in skin if there is a vitamin A deficiency? Because we have read vitamins A is responsible for the cell growth and cell differentiation. Okay, and we have seen that vitamin A, it helps in converting the keratinocytes into the mature epidermal cells. So if these this maturation doesn't occur, so this will be presented at the skin. It will become dry. Okay, it becomes dry because of the reduced serum secretion, and there would be increased risk for the infections. Okay, एक और जो हम skin manifestation हो सकते हैं, that is the follicular hyperkeratosis, and it occurs because of the excessive keratinization of hair follicle epithelium. ठीक है. So in skin you can see the skin in vitamin deficiency, it becomes dry, it may become dry, and they will also be follicular hyperkeratosis. Okay, now you all must be familiar with the disease. I think you might have heard acne. Okay, acne, as you see that this disease is very mostly it has been seen in teenagers now, and it is because of the excessive secretion of sebum from the sebaceous glands. Okay, and इसका reason क्या होता है? Mostly it is being triggered by the hormonal imbalance. So what we do is that in this disease we give vitamin A as synthetic vitamin A, isoretinol. Okay, so what does it do? It just prevents the acne by reducing the sebum secretion. Okay, so sometimes this vitamin A is being given as a treatment for acne. Okay, now this picture shows the follicular hyperkeratosis. You can see so many of, because of the hyperkeratinization of hair follicles, hair follicle epithelium. Because in deficiency, this epithelium will not be maintained. Okay, and the hyperkeratinization will occur, giving rise to follicular hyperkeratosis. Okay. Now, what are the recommended daily requirement for vitamin A? In children, it is 400 to 600 microgram per day. In men, it is 750 to 1000 microgram per day. In women, it is 750 microgram per day. In pregnancy, it is 1000 microgram per day. Okay, and sources of vitamin A, as we know, sources of vitamin A, mostly like dairy products we get, like cheese, milk, yogurt, butter, and these are very good sources of vitamin A. Other than this, we have the colored fruits and vegetables like the bell pepper, spinach, then vegetables. Okay, then we have carrots. They have a very good, very rich source of vitamin A. Then in fruits, papaya, okay, mangoes, peaches, they are very good sources of vitamin A. Okay, now what are the therapeutic uses of vitamin A? As I have already told you that vitamin A, it cures basically the disease which is being caused by its deficiency. So the most common therapeutic use of vitamin A is when there is a vitamin A deficiency, when it is identified. Okay, so in vitamin A deficiency, it has been given, and it has been given in the supplementation as capsules or injections, and usually the dose at 20 to 50 times more than the recommended daily allowances. Okay, and it is being administered in the form of alltrans retinoic acid, or okay. So there is one disease, promyelocytic leukemia. Okay, so it seems to be have a role in this also, vitamin A, because it helps in the proper cell differentiation, in promoting the normal cell differentiation. Now, as vitamin A, it is a lipid soluble vitamin. So if it has been taken in excess, it can also lead to hypervitaminosis or toxicity in our body. Now, normally, food, it does not cause this toxicity. It is the excessive intake of vitamin A when it is being taken as supplement, or being fortified, or we take the fortified food which has been fortified with the vitamin A, can lead to toxicity if it has been taken for a very long time. Why? This we have already discussed. Because they have because being lipid soluble, they can be stored in our body. Okay? And their excretion is also not, they are not being readily excreted. So they have a tendency to build up in our body. So it can cause toxicity. Now it has been seen that the people who are in the Arctic region, they consume excess of liver of polar bears. Okay, so they have been seen to have this toxicity, right? So, or the people who are self-medicating themselves with the vitamin A, they may also develop this toxicity, okay, because of the vitamin A. And it has been seen that when we take the vitamin A in the amount that supersedes, when this toxicity occurs, whenever we take the vitamin A in the amounts that supersedes the binding capacity of retinal binding protein plasma. So at that time only the toxicity appears. Okay? When it supersedes the binding capacity of retinal binding protein plasma, only then it will cause this toxicity. And this is usually being seen when it is 10 times above the recommended dietary allowance. Okay. Now, what are different symptoms of toxicity? The toxicity, irritability, headache, drowsiness, vomiting, and this because of the due to increased intracranial tension. Okay? And they could be swelling over the long bones with pain in the bones. Okay, swelling with the pain in the bones, that is known as bony exotosis, and there could be enlargement of the liver because of its storage, because it may get stored in the hepatocytes, so there will be enlarged liver. Okay. Then it has been seen that higher concentration of retinol also increases the release of lysosomal enzymes, which can lead to the cellular death. Okay. Now, what is the management? Management would be symptomatic. What are the symptoms? That would be patient will be treated as per the symptoms, and then removal of the cause. What is the cause? That cause has to be removed. Okay. So this is all about the hypervitaminosis because of vitamin A. Now there is a term hypercarotenoemia. Now this is hypercarotenoemia. It is like it occurs when there is an excessive consumption of foods which are rich in carotenoids or the beta carotenes. Now, as we know that uh, okay, so in this, what happens? This because we know that the beta carotene, it is being less it is being metabolized very slowly in our body. So what happens, instead of getting converted into retinol efficiently, so it starts because it is lipid soluble. So if it starts storing in the adipose tissue. Okay, so what happens because of this excess of beta carotene in adipose tissue cells? What happens? It imparts a yellow cast to the skin. So this condition we call it as a hyperkeratonemia. So okay, so with this I complete the vitamin A. Now next I will be discussing about vitamin K. The next fat soluble vitamin K. Vitamin K. ठीक है? Now vitamin K. This is actually there are two forms of vitamin K which are being present naturally. Okay, what is K1 and the second one is K2. Okay, now K1 known as phyloquinone, it is being obtained from the plant sources, and K2 menaquinone, it is being obtained from the animal sources. Okay, and also our body, the intestinal flora in our bodies, they are responsible for synthesizing this form of vitamin, that is vitamin K2. Okay, there is one more form, that is K3. Now this is a synthetic form of vitamin K. Okay, synthetic form of, not readily available, but a synthetic form of vitamin K. And as opposed to K1 and K2, it is a water soluble vitamin. It is a water soluble vitamin. Okay, although we are studying it as a fat soluble, but K3 Minadion, we call it as it is a synthetic vitamin K. Okay, and water soluble, and it has a vitamin K activity which is being widely used in clinical practice. So in clinical practice we use vitamin K in the form of K3, okay, which is water soluble, right? And now all the vitamin K, like the compounds which come under the vitamin K, they are being the naphthoquinones, okay, and this is a naphthoquinone, and there is a isoprenoid unit that is being substituted at this carbon atom. Okay, so basically they are isoprenoid substituted naphthoquinones. Now, what about absorption and storage? As we have seen in all fat-soluble vitamins, absorption of vitamin K, it occurs in the intestine with the help of bile acids. Okay, and once they are inside, after absorption, they're inside the intestinal cell, they get incorporated into chylomicrons, and from chylomicrons, then they are being through the lacteals, they reach the to the blood circulation. Okay, and here what happens? This chylomicron then after being converted as a chylomicron remnants by various peripheral tissues, it goes to the liver, taken up by the liver, this chylomicron remnant. Then as per the requirement of the body, if it is being required, they are being transported in the along with the beta lipoproteins in the blood, or they may get stored in the liver. Okay, so this is the similar, all the vitamins, they have the similar metabolism of absorption, of transport. Okay, then what happens? Okay, now what is the biochemical role of vitamin K? Now it has a very important role in blood coagulation. So basically some of the factors like blood coagulation factors like factor 2, prothrombin, factor 7, factor 9, and factor 10. Okay, so they are dependent on vitamin K for their optimal biological activity. Okay, how? Now these, as you know that all the clotting factors, they are being synthesized in liver. Okay, now these factors, factor 2, 7, 9, and 10, they are synthesized in liver in the form of inactive precursor forms. Okay, they are in the precursor forms. So that they can exert their biological action, they need to be undergo a modification. We call that as a post-translational modification. Okay, now this post-translational modification, it occurs in liver. Okay, as been synthesized and being they undergo post-translational modification in liver. Now how this post-translational modification, what modification is required? See, all these clotting factors, they have glutamic acid as one of the residue. Okay, so this residue, what happens? It undergoes the carboxylation. It needs to be undergo carboxylation so that these clotting factors, they can have their optimal biological activity. Okay, so how does the carboxylation of this glutamic acid residue in these proteins it occurs? It occurs through a carboxylation reaction, and in which, and this carboxylation it occurs in the carboxylation occurs at the gamma carbon atom of glutamic acid. Okay, glutamic acid residues, and this carboxylation reaction it requires vitamin K as its coenzyme. Okay, and why this carboxylation is needed? This carboxylation is needed because after uh, these proteins they get these clotting factors they get carboxylated at their glutamic acid residues. They uh, become they become active, and they behave as calcium chelators. Okay, so you can see here in this post translational modification what is happening. The glutamic acid it is getting converted into gamma carboxylic glutamic acid, or glue. We call it as a glue. Glue is getting converted into GLA. Okay, so here you can see this is the reaction. The glutamic acid residue in the protein chain of these clotting factors of prothrombin of clotting factor 2, 7, 9, and 10. It is getting carboxylated. See here, this is the glutamic acid. Okay, this is alpha, beta, and gamma. This is gamma carbon atom. Okay, so here you can see this is gamma. So in the presence of an enzyme carboxylase, which is vitamin K dependent, this glutamic acid residue in these proteins they are getting converted they are getting carboxylated at their gamma carbon atom. Okay, so this is alpha, beta, gamma. So this is one carboxyl group. The second one has been added here. Okay, so in this way the glue is converted into GLA. Okay, the glue glutamic acid is converted into gamma carboxylic glutamate to another amino acid, that is GLA. Okay, so here now these two carboxylic acid they are responsible for chelating the calcium. Okay, in this way they behave as calcium chelators, and thus they facilitate the coagulation blood coagulation process. Okay, by mediating the clotting factors calcium and phospholipid interaction. Okay, so it is very important, and okay, so this is happening. This reaction is vitamin K dependent. Now see how it is vitamin K dependent. You can see here now while this glutamate is getting converted into alpha carboxyl glutamate in the presence of alpha glutamine carboxylase, this vitamin K it participates in reaction in its reduced form. Okay, it is in reduced form. This is hydroquinone form. So what is happening? Glutamate is converted into alpha carboxyl glutamate, while this vitamin K it is getting converted. It is getting oxidized and getting converted into vitamin K2,3 epoxide. Okay, now this needs to be again get converted into vitamin K, and how does it occur? This occurred with the help of an enzyme vitamin K epoxide reductase. Okay, तो वो क्या करेगा, इसको जो oxidized vitamin K है, it will convert it into vitamin K. Okay, here you can see and this happens from here. It is getting converted into 2,3 epoxide is converted into vitamin K. Okay, and here we have the sulfaldehyde groups. There are some groups which are providing this reducing equivalent, and they themselves get converted into oxidized dithiol, and while converting this 2,3 epoxide into vitamin K. Okay, now this vitamin K, then with the help of reductase, it gets where NADPH, it's an coenzyme, gets converted into hydroquinone, and this in this form, vitamin K in the form of hydroquinone, okay, it participates in carboxylation reaction, converting glutamate residues in these proteins into alpha carboxyl glutamate. Okay, so this reaction is very important because they convert these precursor forms of these clotting factors and make them active to participate in coagulation, in blood coagulation. Now, what are the sources of vitamin K? Green leafy vegetables are very good sources for dietary sources for vitamin K, but intestinal bacteria synthesis, it is sufficient enough to meet the daily requirements. So vitamin K deficiency seldom occurs in a normal individual. Okay, so okay, now this deficiency can occur, but now what are the causes for deficiency? What are the causes for vitamin K deficiency? Now, in normal adults, the deficiency seldom occurs because the intestinal bacterial synthesis is sufficient to meet the needs of the body. Now, deficiency occurs whenever there is a malabsorption of, there are conditions leading to the malabsorption of lipids. Okay, like we have obstructive jaundice, chronic pancreatitis, flu, etc. These clinical conditions will lead to the malabsorption of lipids and will in turn will affect the absorption of vitamin K. Okay, then another reason for the vitamin K deficiency is prolonged antibiotic therapy. Okay, because this with the prolonged antibiotic therapy that this prolonged therapy can damage the bacterial flora, due to which the synthesis of vitamin K would be then the supply of vitamin K from this bacterial flora will be lessened. Okay, which can then lead to the vitamin K deficiency. So the prolonged vitamin K and prolonged antibiotic therapy or gastrointestinal infections with diarrhea. There are chronic gastrointestinal infections are there. That person also may have the vitamin K deficiency because of the destroying of the bacterial flora. Okay. Then there is one more cause for vitamin K deficiency, that is the prolonged use of anticoagulants. Now we give anticoagulants in some situations, like we give the patients which suffered from the myocardial infarction or which are having the deep vein thrombosis. So in such conditions, we give the anticoagulants. And but if these anticoagulants in a very high doses and for be given a very high for a very long time, then they can cause the vitamin K deficiency. How? We can see this one of the anticoagulant warfarin. Basically, how does it causes the anticoagulation? It basically is a competitive inhibitor of vitamin K, 2,3-epoxide. Okay, so it competes with this enzyme and thus it blocks the conversion of vitamin K from hydroquinone. It blocks the conversion of this 2,3-epoxide into hydroquinone. So if it is not available, then what will happen? This hydroquinone will no longer be available for the conversion of glutamate into alpha carboxyl glutamate, and due to which the precursor forms of these clotting factors, they will not get activated and they will participate in the coagulation process, so leading to the coagulation failure. Okay, this is basically the mechanism of action of these anticoagulants. How do they act as anticoagulants? Okay, so prolonged use of these anticoagulants may also cause the vitamin K deficiency. Okay. Now, what could be the clinical manifestations of vitamin K? See, now the vitamin K deficiency in newborns is usually being manifested as a hemorrhagic disease, and those infants, premature infants, they are more they are being prone for having vitamin K deficiency, because in them the intestinal flora is not being developed properly, so they become more prone for the hemorrhagic disease because of the vitamin K deficiency. And also they have the lesser stores. These premature infants, they have a lesser stores of vitamin K in their body. Okay, so in this such preterm infants or the premature infants, prophylactically vitamin K is given in the form of Minadion. Okay, otherwise in children and adults, vitamin K deficiency, it is being manifested as the bleeding disorder, like they may be manifested as losing tendency, ecchymotic patches, mucosal membrane hemorrhages. They may have post-traumatic bleeding, nasal bleeding, internal bleeding. In deficiency, the clotting that would be hampered. Blood clotting would be hampered. Okay. Now, how would be then diagnose the deficiency of vitamin K? This we perform a test, that is the PT. जैसे प्रोथ्रोम्बिन टाइम, ठीक है, उसको हम प्रोथ्रोम्बिन टाइम मेजर करते हैं, उसमें क्लॉटिंग टाइम मेजर करते हैं, पीटी एंड सीटी. तो क्या मिलेगा हमें अगर विटामिन के डेफिशिएंसी है, इट इज हैपनिंग बिकॉज देन देयर वुड बी अ प्रोलॉन्गेशन ऑफ प्रोथ्रोम्बिन टाइम. Okay, and in clotting time also there will be a delay in the clotting time. These are the characteristics of vitamin K deficiency. If there is a vitamin K deficiency, there would be a prolongation of clotting of prothrombin time, and there would be a delay in the clotting time. Okay. So now, what would be the daily requirement of vitamin K? This would be like 50 to 100 microgram per day, and it is very less because most of the vitamin K is being supplied by the intestinal flora. Okay, so there are conditions in which the hypervitaminosis K can occur, and this usually occurs when a very large quantity of Minadion is being administered for a very long time. Okay, when excess, when it is being given higher doses for a very longer time, only then the vitamin K toxicity can occur. Okay, and then what could be the clinical features in that? Hemolysis, hyperbilirubinemia, nicterus, and brain damage. Okay, so these are the manifestations of toxicity. So this is all. I'll be now completing these two vitamins, like vitamin K and vitamin A. Okay, so hope you have understood. Vitamin A, I think, would had taken a longer time. So if you have any query, you can please write to me. Any questions? Do you have any questions? Okay, I'm waiting for some time. If you have any questions, so please write down. Okay, one student has written HRE. The full form of HRE is hormone responsive element. Okay. Any other question? बच्चे mechanism of Rawls function, बच्चे ये तो हमने भी पढ़ा ही है सारा. What is the mechanism? Amisha, what do you want me to summarize? What in vitamin A or vitamin K? Amisha? Okay. see summarize इसमें mechanism of rods function is that कि जैसे rods हैं, rods they have a visual pigment in them, rhodopsin. Okay, so what happens when light falls on these rods, the rhodopsin, which is basically rhodopsin, it is a protein which is conjugated to which is bound vitamin A, ठीक है, and this is in the form of 11 cis retinal. Okay, तो क्या होता है कि जब light जो है when the light falls on the rods, so this rhodopsin of protein it gets bleached. ठीक है, bleached का मतलब है कि जो 11 cis retinal है उसके अंदर जो 11 cis retinal it gets converted into all trans retinal. Okay, now since this all trans retinal it is bound to the protein opsin, so it because of the change in its structure it causes a conformational change in the protein opsin. अब उसकी वजह से क्या होगा कि जब कन्फर्मेशनल चेंज होगा तो जो बाउंड 11 सिस रेटिनल है वो उसके से डिसोसिएट हो जाएगा, ठीक है, इस प्रोटीन से डिसोसिएट हो जाएगा. अब डिसोसिएट होने के बाद जो ऑप्सिन है जिसमें ऑलरेडी कन्फर्मेशनल चेंज आ चुका है, दिस ऑप्सिन नाउ विल activate a G protein which is being present, okay, G protein which is being bound to the cell membrane. Okay, so it activates this G protein, and this G protein when it's get activated, it this G protein will now further will activate enzyme phosphodiesterase in the rod cell. ठीक है, उससे क्या होगा, जो cyclic GMP channels थे वो जो है cyclic, it will then affect the ultimately it will affect the permeability of the cell membrane of rods. Okay, so falling of light causing the bleaching of rhodopsin, and because of the bleaching of rhodopsin, because of various through various biochemical reaction, basically it is causing a change in the permeability of the rod cells and making the rod cells more hypopolarized. जैसे hypopolarized वो होंगे तो क्या होगा, they will be a trigger for the generation of nerve impulse. ठीक है, वो nerve impulse then will be transmitted to various cells in the neurons in the retina through optic nerve to the brain for the visual perception. Okay, or you need to remember कि आपको याद रखना है कि वो हो कैसे रहा, how does this changes being brought about by the rhodopsin, कि rhodopsin से cellular change ये जो है cell permeability जो है उसकी how it is changing the cell permeability. Okay, so this is all about the role. ठीक है, and then this is the stimulation, how it is stimulating the rod cells. अब इसका कैसे होगा, shutting down of stimulation कैसे होगा, दो mechanism से हो सकता है. एक तो first is G protein which was being activated, it gets inactivated, ठीक है, and number two, association of 11 cis, again the association of 11 cis retinol into association of this to opsin. Okay, तो दो तरह से है, two mechanism they will be responsible for the shutting of the stimulation of rod cells, right? So is it clear? Okay, so if you have any problem, so we can discuss in the small group discussions. Okay, so now I think I'm just stopping here. So and then we'll meet tomorrow to cover our rest of the vitamins, vitamin E and vitamin D. Okay, so thank you so much. Thank you to all of you. धैर्य इसको बेटा कैसे करेंगे यार, यार कम आ रहा है धैर्य।